This is crucible tongue. Actually, crucibles are small structures, or you can say small operators, which are used for heating chemicals. Crucibles are more strong than glass and more resistant to chemical, so you can heat them at very high temperature. So these crucible tongues are according to how you can hold crucibles easily. I will show you crucible in further diagram, in further slides. Test tube holder, obviously it is for holding the test tube. If you want to heat up any sample of your test tube, you can use these kind of holders. Or for example, you're adding H2SO4 in your test tube, so you can't hold that test tube directly in your hand. Otherwise, what will happen? It can burn your skin. So we use these test tube holder. Test tube brushes are used for washing the test tube. Those test tubes which are made up of glass, high quality glass, are reusable, or so for washing we use these kind of test tube brush, because test tubes has small diameter. So these kind of test tube brushes are inserted inside the test tube for cleaning purpose. This is scapula. If you want to take some chemical from the stock, for example in laboratory what we have different bottles in which different chemicals are placed. Let's In this bottle it is sodium chloride. In this bottle it is glucose. In this bottle it is sucrose. In this it is copper sulfate. So what we need to take some chemical, because when we are doing any experiment or any test we need small amount of chemical, right? So by the help of scapula you will take chemical. What is precaution for using scapula? Before using scapula, you will cover it with the help of foil paper. With foil paper. So now what you do? For example, you cover that scapula with foil paper, and from this bottle, you remove some amount of sodium chloride. Now what you will do? If you want to take some another chemical, for example, you want to take copper sulfate now. What you will do? You remove that foil paper which you previously attached. Now put new foil paper on that scapula, and then take copper sulfate. So it will prevent mixing of chemical in your laboratory. Yes. Whiteboard is also not in front of you. Right now I am showing you from whiteboard. Wait, just wait a minute. I don't know why it's normally disappear from screen. Okay, just inform me whenever this kind of situation take place. So we are on scapula. Look here. Wait. Just a minute. All these we are completed. All these topics are done. We are on scapula. So it is used for transfer solid chemicals. I already gave you examples. For example, sodium chloride, copper sulfate. Copper sulfate is of blue color. Sodium chloride, which is commonly known as salt, is of white color. Then we have glucose, which is also of white color. Sucrose, which is also of white color. We have H2CO3. All of these are of white color. So I am giving you, okay, so we are right now we are studying scapula, which is used for taking some solid chemical. What is main precaution used during using spatula? You had to put foil paper. Yes, still not you are able to view PPT. What happening here? Wait, just wait there. Is it okay? So this I hope you understand for what purpose copula is used now. We can move. I hope you understand for what purpose copula is used. You understand it? Yes, so we can move further to the next slide. So this is thermometer. You know, for example, we are doing some chemical reaction, or you're performing some test. So what happened? Some we need to heat some chemical reagent, right? For proper reaction. So you want to heat only up to 50 degrees Celsius. So in the test tube or in your beaker, you will put this thermometer. So as the reading come to 50 centigrade, 50 degrees Celsius, you will stop heating. And thermometer consists of chemical known as mercury for the reading purpose. This is forceps. It is used for holding the samples. For example, you are dealing with some tissue, so you can hold that tissue with this forceps. This is medicine dropper. Dropper is for what purpose? Dropper is used for taking small amount of liquid and adding it to the some another chemical or mixing whatever. Test tube rack used for placing test tubes. You can see here these rounded structure. In these you can put your test tubes. 1, 2, 3, 4, 5, 6. 6 test tube can be placed in this test tube racker. We have variety of test tube rackers. Some are made up of wooden, some are made up of plastic, different kind. Some can hold 10 test tubes, some can hold 20 test tube. This is glass stirring rod. Stirring rod is for stirring the liquids so you can mix liquids properly. And this it is normally of up to it is normally 20 centimeter or 30 centimeter in length. So you can stir chemical easily and there will be less chances of slippage, spillage as well as less chance of any burning or any. Just a minute. Just wait a minute. All these topics are completed. Forceps, medicine dropper, test tube cracker, glass stirring door. This is Bunsen burner. In laboratory, these burners are used for the purpose of heating. It is directly attached to the gas supply. We will place these kind of just give me a minute. I will show. These type of clay triangle are placed on this Bunsen burner, and then we place our beaker or our flask on Bunsen burner. Okay. So in laboratory, you should take care of the gas supply. Whenever you completed your experiment or practical, you should close the gas supply from the source. This is retort stand. Sometime in chemistry laboratory, we use titration flask, or for example, we need continuous supply of liquid or any chemical for a long time, then we use these kind of retort stand with burette. We will place burette in this retort stand by help of C clamp. Let me show you a collective diagram, then you will understand it. What is this retort stand? What is clamp stand? And what is burette? Just a minute. Yeah, dear, look here. Look, this is your clamp stand, this black one, and this rod is attached to the clamp stand. This is burette, the plastic tube which you can see. And this holder, this is C clamp which is holding this burette. And this is top cork. This blue one is top cork. For example, you want to stop the flow of liquid, you will close it. You want to on the flow of liquid, you will rotate it on opposite side. Flow of liquid will start. So these are three things. This diagram is covering your three equipment: retort stand, burette, C clamp. Done. Retort stand. C clamp also done. It is used to hold or secure object to the retort stand. I hope you understand this. This is burette. It is a vertical cylindrical piece of laboratory glassware with a volumetric graduation. What is mean by volumetric graduation? On burette also you can see, when you see diagram later on, you can see there are marking like 1 ml, 2 ml, 3 ml, 4 ml. So these are the graduation means marking on its full length, and a precision tap, that tap which I explained you. If you open that tap, flow of liquid will start. If you rotate that tap on opposite direction, flow of liquid will stop. So that tap is also known as stop coke or precision tap, used to dispense non amount of liquid. For example, you only want to add 1 ml of liquid, you will add, and you will see 1 ml is added, you will close the stopcock again. It is used in chemistry for titration purpose generally, or for example in any biological experiment you need continuous flow of any liquid. So then we will use this kind of burettes. Wire mesh. Wire mesh is usually used for filtration purpose, or you want to place something on Bunsen burner, then you can use this kind of wire mesh. This clay triangle this clay triangle generally placed on Bunsen burner for placing other equipment like glassware, other glasswares like beaker, flask. This is a crucible which is used for heating purpose of chemical. It is very hard and non-reactive. It will not react with chemicals which you are going to mix in your laboratory. Funnel, when you want to add liquid in burette, you can't add that liquid directly. Why? Because diameter of mouth of burette is small. So you need these kind of funnel. through you will place funnel on the mouth of burette, then you will add liquid. Filter paper, it can be used for filtration purpose, or normally we can perform some kind of experiment by using these kind of filter paper. Mortar pestle, mortar pestle for grinding the solid substance. For example, you have some tissue, you want to grind that tissue, you will use these kind of mortar pestle. Spot plates, these process small depression. For example, you want to do blood testing, you want to know what is the sample of blood, ABO, then you use these kind of plates in which you will first add blood sample and then you will add antigens. We will do blood group, how blood group testing can be performed. Evaporating disk, for example, you want to evaporate some chemical from the mixture, you can heat things in this evaporating disk. Petri disk is used for culturing of cells in the laboratory. Culturing means grow, for growing the cells in laboratory. Watch glass, it is used as cover. For example, you want to cover some any beaker or want to cover petri plate, then these kind of watch glasses are used. And your pipette got finished. So, are you able to understand these equipment or not? At least you understand some points, what kind of equipment we normally see in our lab. Our next topic will be cleaning of glassware. Okay, so we will continue tomorrow. Not tomorrow. I think you have lecture with me now on Monday. So we will study these topics. We will study cleaning of glassware on from Monday. Okay.